to cloud. Yeah. Okay, go ahead.
Oh, sorry. All right, hi everybody. So some of you guys might know me by now. My name is Thomas Schmidt. I am working at the NIH, specifically on EPR and in the laboratory of NIH-Xplor. And so how should I say? So we have lately been interested, obviously we do EPR, and it might not be a big secret, but I'm really a one-trick pony, in which case it's just that I do DEER and apply it as many systems possible in different variation of such. Now, lately we have become interested in calmodulin. I mean, calmodulin has been studied. Everybody knows it. Everybody loves it, and many folks. There remains a question of how a calmodulin behaves in the calcium-free bound. How does it bind to its substrate? And we have been using rapid freeze quench system to specifically look at all those questions. And in this talk, I announced that I'm going to talk about the calmodulin binding to its substrate. I'm still going to do 4 or 5 slides on that. But because my recent paper got published on the substrate free state and the resolution of different substates in the calcium loaded state, I'm going to talk about that largely. So sorry about that. Okay, so again, DEER, as well as many other structural biology or biophysical experiments, treat the idea of a structural information. Now, structural information is great, and it pushed the limits on so many fronts, including drug discovery. However, as time goes on, I feel like we need to sway into different areas as well. And one of those areas is kinetics, specifically equilibrium kinetics and non-equilibrium kinetics. Now, just to quickly introduce the concept, what is equilibrium kinetics? Well, equilibrium kinetics is really, if I take a protein and I put it in solution, it probably might have different substrates, might have dimer, trimer, God knows what. We want to see how those different equilibriums are. So how are they exchanging with each other? So the system is not disturbed. It's just simply lying there. And one can imagine that one just simply takes a snapshot of a running horse. You simply take a snapshot and you see what the horse is doing. And the concept becomes that, well, I see the horse. And if I don't know whether there's a horse, it would almost appear the horse can be flying. Of course, that's logical. But also in another picture, we can see it landing, but we don't know how the sequence of that horse is related. Is it going forward? Is it going backwards? I don't know. So therefore, the concept of non-equilibrium kinetics comes in which we actually now look at the different conformational landscape of a protein. And by doing such, one can imagine that if we now have our protein in the equilibrium and we introduce it a disruptant, be it a substrate, be it temperature, be it pressure, we can now disturb the protein behavior and then take a movie as it unfolds. And what it becomes is that I can see the horse now running. I can see its behavior. I can go actively on the racetrack and see if the horse is flying or just running and how it runs and how it lands. And I think this is really the next frontier of structural biology, to really resolve those different conformations which give rise to this kinetics. Now, to do that, obviously, we're going to use a DEER experiment in EPR. And simply what it is, I'm just going to introduce it because I know many people are very familiar with it, is that I have a protein which exists in multiple different conformations. And most likely for EPR spectroscopy, we already have a pretty good understanding about some structural information, like local structural information. But we don't know how the conformation in the global aspect is and how this conformation is changing. And so if we want to know how a structure looks like from all those different conformations, we normally utilize surface exposed cysteine and covalently bind a paramagnetic label, which mostly is the MDSL label to our protein. And one can imagine that once we have two MDSL labels on our protein, it will become like a cone-like shape, as previously described by Eric Houston. And if that is the case, we can now utilize our DEER experiment to measure distances. And again, we would have a time domain. We have our signal, which is convoluted with a background. And if one uses now various mathematical tools, one could now extract the distance between those two parametric labels and then further restrain the conformation against those distances or distance. And the final result will be that we restrained our protein. Now, in our lab, we do that and everybody does it. I believe many of you guys are quite familiar with it. However, further we implement isotope labeling. Isotope labeling, what does it mean? It does not mean we're using radioactive materials. It just simply means that we use deuteration or site-specific protonation or other isotopes, which will have an effect on the nitroxide label, as I will explain shortly. Subsequently, we also use an effect. I don't want to say it's a full sequence, but it's an effect called TM-edited DEER, in which we follow a relaxation profile of distances under the influence of isotope labeling. And all of that will be then taken together in a global analysis aspect as pioneered by Eric Houston in the DD or DEER lab, something in the DEER lab program and from the Jeschke lab, or the 2D SVD program from Matteo Srivastava. So all of that together would give us an extra enhancement of resolution, what's going on during the conformational exchange process, but more to it later. Now, my background is NMR, and we all know that NMR is slightly ahead of EPR, sadly. And part of the reason being simply that NMR can look at chemical shifts and therefore can resolve specific amino acids and specific behaviors, and then subsequently assign the chemical shifts due to specific sites in the protein. Now, I believe, however, that EPR can catch up, and we are now in the maybe 88, maybe 1990s. However, since we are now able to use isotope labeling in NMR, we can, specifically method labeling, we can now introduce artificial chemical shifts. Now, that does not mean necessarily chemical shifts from the NMR principle, but we can look at the relaxation behavior of our radicals in a localized chemical environment, meaning we can see if an electron is specifically coupled to specific nuclei and also resolve the distances at the same time. And we largely have shown that by using method labeling. Just a little introduction to this principle as it becomes more important later on is that, for example, if I have my model protein of choice, protein A, I have it labeled at the N terminal and shown in red, and C terminal shown in orange. Now, the N terminal label will split into two rotamers and therefore gives it a typical two distance profile, as one can say here, as a distance at 31 angstrom, another one at 39 angstrom. Now, if I have protons located only to one rotamer population, meaning that L-leucine 35 is in very close approximation to the B rotamer population, this coupling between the electron and the proton will cause a rapid decay of this distance compared to the other one. And how does that look in practice? So again, if my proton population is in close distance to my electron, so here shown in this area. Now, because in close proximity, my distance will rapidly decrease in comparison to my other distance population. And that simply is due to the proximity. And therefore, we can actually assign this distance simply to the N and C terminal. So this distance comes only from B to orange, whereby the other one comes from A to orange. And this is very easy principle to follow. As one can imagine, the simpler Haneke T2 relaxation. So one of the spins is shown in red if it's close to a proton. And if it's simply in deuterated environment as all the other electrons, it will have a T2 profile, which is shown in blue. And yeah, so just keep this principle in mind, and we will follow up on that later on. Now I need to introduce another principle, and this would be the rapid freeze quench system. In a rapid freeze quench system, which was pioneered by Ken Thurber, Ca2, Young, and the TICO group overall, we are using cold copper disc, and we rapidly mix two proteins. Now, in our case, because we're going to work with calmodulin, we have calmodulin mixing with the substrate, which is MLCK. And as I injected on the copper disc at a specific time frame, mixing time frame, I can resolve now specific steps of folding or refolding as a bound state form. So this is called the reaction time. Meaning, if I mix it together, I inject it on my disc, it freezes in time. Now, if something freezes in time, this is a huge benefit now, because compared to liquid state, I have infinite time now to measure and extract distances. This is great. And obviously, it's the same true for solid state NMR. Now, I introduced two concepts there. I introduced the TM filtering and as well as rapid freeze quench. And again, this talk will be on two projects. The first one is just the calmodulin kinetics, so mixing calmodulin with a substrate and freezing and looking at conformational changes as it binds a substrate. And the major part will be actually simply on the calmodulin itself. Anyways, so from now on, we're going to go on from calmodulin kinetics and actually the bound state formation. So what do we do? I already introduced many principles. However, just to comprehend it, the big steps in looking at protein kinetics, at least by EPR, is obviously we have our substrate, we have our calmodulin and Ca2 bound state. And what techniques are we going to use to specifically resolve each individual time reaction time coordinate step? So first, we're going to enhance the signal. So meaning, how do we enhance signal? Simply by protein deuteration. So protein deuteration means that I'm not relaxing my signal significantly anymore because we don't have protons present initially. And also, so that gives me a big boost in protein and in signal. So in DEER signal for that sake. Then we're going to do a time resolution. So we resolve each individual freezing step by using the rapid freeze quench system by simply injecting on the cold copper disc. Now, if I do that, I already, as an EPR spectroscopist, we already know there's going to be a bunch of distances which are just smack on top of each other. So we're going to have a spectral crowding going on. And we need to have some spectral resolution and some enhancement on that. And for that, we're using proton relaxation enhancement, meaning even though we initially have everything deuterated, we're going to specifically introduce protons. In this case, we use a substrate which is protonated to enhance, to isolate distances which are overlapping with each other, meaning that there are going to be some distances which are decaying in the T2 dimension compared to the other distances. And therefore, we're going to get some spectral resolution enhancement. And at last, what you're going to see is that we have a data analysis. In this case, we simply do a global analysis where we're going to have 3 dimensions, or pseudo dimension, I should say. That is obviously the distance, so the time dimension. Then we have the reaction time dimension, so with the rapid freeze quench system. And at last, we're going to have the T2 dimension, meaning that each individual distance will have its own T2. And when we analyze the data, or in case we fit it, we're going to see that each population will change due to its T2. That will give me a resolution of 3 dimension or pseudo dimension, which is a big boost. So how does it look in practice? So I'm just going to have that shown here. Again, we have our substrate, which in this case is protonated. So keep in mind, this is protonated. And we have our calmodulin where our paramagnetic labels are on the anode C-terminal side. Now, as our protonated substrate encounters that deuterated calmodulin, the distances associated in close proximity to its substrate will decay rapidly. And this can be seen right here. So again, we take our mixer, we inject it on the copper disc at different time points. And if I just take the initial time step, I'm just gonna see like some blob. So it's not very well resolved. We don't know what's going on. But now if I apply the T2 filter, because some distances, early distances, which are in close proximity to the protonated substrate will decay rapidly, I can now use different 2D programs. In this case, I mean, actually, in this case, we used Eric's global analysis part, where we can now see that some of them are decaying more and more. Now you can see that initially, yes, they're like almost the same height, but as time goes on in the T2 dimension, we can see that the initial peak at around 22 angstrom is rapidly, population is rapidly decaying and diminishing. And this is just due, and from that we can already tell that this distance is associated with a conformation that is wrapped around the protonated substrate. So this is very great. And the analysis again can be done with global analysis, can be formed with different softwares. Really, I think. In our case, we really just pushed it with the global analysis idea. With the dimension, again, just to keep in mind, we have the simple, like everybody knows, DEER time dimension, where we add time to distance. And then we have the T2 dimension, which I introduced here by having that concept of having a deuterated protein with a protonated substrate bound. And at last now, to relate the different freezing points in the reaction times, okay, we need to introduce a kinetic model. So now, this is fun, this is great, this is really cool stuff, because now we can test different kinetic models against our system by Gaussian modeling. This is really great, because now I can take, again, we have our different components present here. Now we can take at some time point, in this case it was 1.5 millisecond reaction time, and we can use the T2 dimension. Again, you see the decay of the short distance, whereby other ones decay differently. And now we can fit it to our T2 models. And now if we do that for a bunch of different reaction time coordinates, so 1.5 to 25 millisecond plus an infinite time point, we can now fit a simple linear model, a simple branch model, or this an advanced branch model to it. And we can really just see how the chi-squares. I mean, this is all really what it comes down to. We just fit it in a global timescale. We have our 3-dimension, everything should fit perfectly and we're good to go. So in a linear timescale, you can see that obviously it doesn't fit very well. If we look at it, please follow just the lower part. You can see it really doesn't fit well. If I do the simple branch model where calmodulin goes with a substrate to an intermediate A, then to another intermediate B, and the final state, which is the fully compact crystal structure C, or it goes from A to C directly, it also doesn't fit actually at all. It's almost there, but it doesn't match the early time points very well. And at last, we have now our last complex branch state where our calmodulin is mixed with our substrate and it goes directly to two intermediates A and B, which it comes down to that it's either it interacts, the substrate interacts with the N-terminal side of calmodulin or with the C-terminal side of calmodulin differently. But by A star can go to another intermediate B and final state C, or A goes directly to C. And if we use that, you can really see it. It's a great fit. It fits very well, and the reduced chi-square becomes very nice. So, of course, to finalize this concept, we obviously didn't just have EPR data. We also use solid state NMR data. And I'm not going to dive into the solid state NMR. I really just want to focus on the EPR part. But what you can come down to is that you have our calmodulin, which is labeled on the N and C terminal side. which both of them have actually affinity for our substrate. And they can both bind to each other. And as we resolve them subsequently, during the different rapid freeze quench times, we can see the evolution of that final compact complex structure occur. But in the conjunction, both EPR and solid state NMR proof showed that that our substrate actually initially can interact with both the N-terminal site or C-terminal site. And from there on, it can actually form an intermediate. So from the N-terminal site, initially interacts, random coil interacts with our folded N-terminal site. And then the C-terminal site will fold onto the interaction and causes a helical conformation on the N-terminal side of the substrate. Now, due to this complex, it will also introduce the C-terminal side of the substrate to form a helix as well, which gives rise to this conformation. In case there's interaction of the C-terminal side, it rapidly goes to the final complex. Now, this is very fascinating because that really drives the point that the recognition, the folding occurs after binding. So there's no alpha helix forms in the substrate already early on. It actually folds upon binding. So this was really great. And it really pushes the idea of Peter Wright further. Now, I'm going to shift the gears a little bit here. It's still going to be calmodulin. But now this is the paper what we recently published, and I'm pretty happy about it actually, in which we look at the conformational substate of calcium-loaded calmodulin. Meaning, how does the calmodulin look like? Even though in crystal structures, it looks extremely elongated, so it's similar to this conformation. But from NMR, we have already had quite a good ideas that that might not be the case and that it might be some other conformation present, minor conformation. And that really plays into the idea that what is the conformational plasticity of proteins? And obviously many people, Dobson and Copless, have pushed that idea of an energy landscape where obviously there are different minor states occurring, but there is obviously the lowest energy state. And in case of calmodulin, that would be the lowest energy state would be the elongated structure. And now we are obviously, the idea is that we use rapid freeze quench as well as nitrogen freezing and simply just putting in -80 freezer freezing, so meaning different freezing rates to identify minor population or conformation which deviate from the crystal structure. Now, of course, we're not the first one to have the idea of utilizing rapid freeze quench. However, I think previously groups have failed to really explore that energy landscape and see what they can do with it. I think the first one to really propose the idea was actually from the Fried group, where they used the similar system of a copper freezing disk and look of how the distances change if one freezes or compared to liquid nitrogen. And this is shown here in black and green. And simply in the paper was stated that the conformation of the rapid frozen system is significantly wider compared to the liquid nitrogen frozen. In a subsequent paper, like we actually published later on, we also saw a similar effect where we had a very sharp distance if frozen liquid nitrogen, but if rapidly frozen, the distance expanded and became wider. In this case, we initially just disregarded it and said it is how it is, but I think it's worthwhile to explore more. And therefore, we use our calmodulin system. Now, as I mentioned before, crystallographers have significantly shown over God knows how many structures that calmodulin is really a dumbbell system where we have the N and C terminal side, shown here, the N and the C in a fully expanded helix. So they are just really elongated. However, both single molecule FRET, mass spec, and NMR have shown that that might not be the full truth. They have shown that in this helix, that connection helix, there are a couple of residues right in the smack in the middle that are a little bit more dynamic and therefore can observe different conformations. Minor, but significant conformations, which probably allows the protein to fold onto a substrate subsequently. In NMR, obviously there's PRE studies by the Chlor group, Nick Anthis, and also in the Bax group, they have shown it through the model-free analysis thanks to NOE, T2, and T1 analysis experiments. However, in our case, the idea is that we take our calmodulin, load it with calcium, we put it in its energy landscape, and we're going to freeze it at different times, meaning -80 freezer frozen. So we give it a lot of time to relax. Liquid nitrogen frozen, we give it a little bit less time to relax. And rapid freeze quench, we're hoping to just cut it off right before it even can start relaxing. And obviously, if one wants to compare it to crystallography, well, we would wait for hours and days. So it has a long time to become in its most energy stable conformation. So how does it look like? So I'm just going to give you guys a snapshot and then we go into the analysis. If one thinks about the rapid freezer quench frozen state, we get obviously beautiful DO traces. We get, as expected, a significant background function. And if we fit it, as I will show later on, we get a very nice residuals. Liquid nitrogen frozen, we get a better background, of course, and residuals still relatively good. And rapid freeze quench, we get an even better background, and the residuals are wonderful. So the freezing rate, what it comes down to is really that the -80 freezer is the longest, liquid nitrogen is the intermediate, and rapid freeze quench is the fastest freezing rate, meaning that if one has a concept of energy landscape, rapid freeze quench really captures the most of the substates present. Now, before going more into the data, I want to introduce the idea that we recorded each frozen sample was recorded with a variety of T2 cutoffs, meaning Ca2 length, even though the acquisition time is always around 7 microseconds. Where we are recording it is different. In this case, we record 10 microsecond, 50 microsecond, 20 microsecond, 25, 30, 35 microseconds. So the Ca2 gets longer and longer. And the reason why we can do that is because the entire sample is fully deuterated. If we would do a protonated sample, we cannot do it because we would have a T2 relaxation effects, and that one cannot do a global analysis on that. And so why did we do that? Why do we record the experiment at multiple Ca2 periods rather than actually recording it very, very long on one Ca2 period? And simply the concept comes in if one does like outlier detection. So if you take your DO trace, you can do it with your own sample, actually. You take your DO trace and you run your, probably in this case, it's better to do a global analysis. And you're looking for outliers. You just follow principles of spectroscopy. You're looking for outliers in your time trace. You will find outliers which significantly have an impact. In this case, it's fitted to Gaussian, to your centers of your Gaussian, your width of the Gaussian, amplitude, lambda, everything is impacted by a couple outliers all the time. So in this case, I have the trace. You can see that one of them is very high up for center. It's just consistent through it. So those are outliers. You can actually detect them very nicely. Now, how do you deal with outliers in DO data? How would one deal with it? So it's a big tricky, right? Of course, you would think, well, if I just recorded over and over again, I could probably average them out. Now, that might be true. However, maybe it's an instrumental artifact. I don't know. We don't know necessarily. Even though the data looks very good, that outlier is huge. And we believe actually removing that point is not a good idea either, because that outlier still contains information. So our principle was that rather than recording one time trace or one Ca2 period for very long, we would just do a global analysis of many Ca2 periods. So we're shifting the acquisition window across the Ca2 period time trace, Ca2 period times. and then just fit all of the traces in a global fashion, which can be obviously done in a model, in a global fashion. And so just that you can see it. In this case, it's a global fit with DEER Lab, where you have all of the traces fit together. And if I do it individually, you can see just the amount of variation that occurs. So I think there's a good chance that that is due to outliers or just simply noise in this end. So I will continue later on with that. Now, when you have your time trace, what do we fit our time traces to? Now, of course, we can simply fit it to Gaussian, and that's all good to do. Or I can do a model-free program by DEER Lab, and that will also be fine. However, and our idea is that, well, we really want to compare it to simulations or like a clusters of structures. Now, we don't fit it anymore simply to a Gaussian. We actually fit it to a known Gaussian or known distribution, and we should simply just need to give the distribution a population. That's distance distribution a population. In this case, we were lucky. Lucy Delmotte from Sweden actually published previous simulations on calcium-loaded calmodulin. And she has specifically clusters which we can simply take and use an in-house developed Rodemer library, which is a spin-off from the Jan Smiler's DEER library, where we modified it slightly to fit it more with NIH-Xplor principles. And if one does that, we can get actually a structure library a distance library, which is based on conformation. So those are all the different structures which are present in the simulations and the clusters. And we simply just use the distances evolved from those conformations to fit the time trace. And we want to see how well we can fit it actually. And we want to give them populations. So this is great because now what we're going to do is we're going to use our time traces. So again, our different Ca2 periods in the DEER trace. And we're going to use our DEER analysis or DEER Lab for using a model-free fit. So again, that allows us to do global fitting, and we can do a model-free fit with it. So that does not assume any idea of what conformation they are or anything. So we literally just feed in the data, and we're going to get a fit out of it. Now, in the next step, we're going to use the DD principle, but instead of just fitting Gaussian, we're going to actually fit a distance library based on conformations to the time traces. And the idea is how good can we fit it, but not just simply how good can we fit the time trace data, but how different is going to be the model free compared to the model fit. We're going to do that for all of them. In this case, and what we can come up with is, again, so just to show you the principle, here is the DEER Lab fit. So again, all of them, the residual look very clean. Then the DEER Lab global fit, again, all of them become very clean. And here is the global fit based on the MD clusters. And all of them are very nice residual, but how does it compare with each other? How do this distance distribution compare? Again, if you just look at the individual distance from DEER Lab, they become kind of all over the place. We don't really know what it means. But if I now do the global analysis and I overlay the model free fit by DEER Lab and the output from the global analysis by our model fit, so based on our MD clusters, you can literally, I don't believe you can see the difference. And the error is in there because of the global analysis, the error becomes very tiny. You can really, I don't think one can see actually a difference in the overlay from the blue to the red. You can see they're almost perfect with each other. And so if one does that, so again, we have our conformation, we have our distance library based on all those MD clusters, and we do our model fit, we can actually now get the population of how much do we need from each of those clusters, and look how they change over time, and we can isolate them. And what it comes down to is that if we do the simple freezer method we have a relatively higher population of our elongated state, which one can believe it's from the crystallography. So obviously, we give it more time to relax, the population becomes bigger. If we do intermediate freezing, this population is just significant, like a little bit decreased. But now we do the rapid freeze quench system, where we rapidly freeze it and try to capture as many substrates as we can. In other words, you can see that this one is now significantly smaller, so it decreased by 15%. And I know the number don't look very big, but if I fit the same population to those time traces, I would not get a good fit. It would be very ugly. And you can also capture different substates, which are now coming up from those conformations. Now, of course, now the first thing what you're going to say, well, Thomas, I mean, yeah, you know, EPR is great and all that stuff, but does it really capture what's going on in the liquid state? Because you're adding, you know, 20% glycerol in this case, you're freezing it in solid, and it's quite different now, isn't it? And you're right. So we need a comparison. We need a method to compare it to. We need to find out if what we are seeing, if we see those substates which we identified, can reproduce data that is present in solution. And so as everybody sits at home and does Scrabble, so did I, and EPR was turning the letters around, and the next thing which I came up with is this anagram PRE. Now, PRE data on calmodulin, calcium-loaded calmodulin, tagged with MTSL is already published. So all what I have to do is take my conformations and utilize the paramagnetic label which is present to recalculate what are the PRE profiles and then compare it to the experimental PRE profiles. And that's exactly what I did, because we all know they are pretty much brothers between each other. And so we took, for example, the elongated state. And if I calculate the PRE profile, the experimental one is shown in gray, and the back calculated PRE profile is shown in red and blue, I will see that those don't give any contribution. That makes sense, because PRE is only sensitive to a very close compact state, so very narrow distances, up to like 20 angstrom maybe. Now, if I go on and I take other conformations, for example, so let's say for rather more compact states, for example, number 11, if I take this conformation now, again, I do the back calculate PRE, I can see that I can capture part of the experimental PRE profiles very nicely, if I may say so. Now I'll go on to number 12. Now, number 12 is even more compact. And lo and behold, I actually reproduce that very big spike by this conformation and some of the C-terminal tail right here. And in 17, again, we reproduce that big spike. So if one goes right through it, those conformations actually reproduce the PRE profiles from the liquid state NMR conformation. So this really gives it a lot of checks and balances right there. So we're pretty comfortable to say that those captured distances mirror the substrate which are present even in solution NMR. And just to recapture the entire setup, we wanted to look at what substrates are present along the energy profile of calcium-loaded calmodulin, and how does it behave for that sake. And for that reason, we obviously use different freezing methods, where the fastest one is a freeze quench that will capture the substrates, the substrates shown in the distances. And we can compare, we can back calculate the time domain as well as the distances, obviously, based on an underlying distance library arising from MD calculation clusters. And this is shown here. So what we're really saying is calcium-loaded calmodulin have multiple substrates. It's not the crystal structure as one believes it is, but we have developed a very nice method that we can actually look at them and directly compare to MD clusters, which I think is very unique. And so I think one can easily implement this idea to different protein folding, distance assignment, protein kinetics, and we can really push on from there. and see where it ends. So at last, I want to say thank you for everybody for listening, and thank for all future, past, present collaborators. If you find yourself here, great. If you're not, please contact me. We can probably get something going. And thank you for listening. Please ask questions if you may. Okay, hello. Thank you, Eric. All right, well, do you guys have any questions? Please ask away. If not, I can point to pitfalls if you guys want. All right. All right.
Questions for Thomas? There's chat. Oh.
Yeah. All right.
Okay.
All right, that was easy.
All right, then let's thank Thomas one more time. It's really wonderful work.
Thank you. Thank you.
Yeah, we'll see you in a couple of weeks. See you next month in Colorado.
Yeah, all good. I will see you guys there.
Okay. All right. Let me end the meeting and we'll move on to some other work.
Sure.